GRGS pathway, adenylate cyclase pathway. Again, we have membrane, yeah. We have G protein couplet receptor with alpha, beta, and gamma subunits, and GDP molecule is bounded to alpha subunit. As we have binding of a ligand with GPCR receptor, we have the attachment of alpha subunit and substitution of GDP to GTP, and activated alpha subunit activates adenylate cyclase. This adenylate cyclase is responsible for conversion of ATP to cyclic adenosine monophosphate, and cAMP can activate different protein kinases again. Activation of different protein kinases, especially protein kinase A. This causes phosphorylation of amino acids or proteins, and as a result, activation of different molecules. Another way is that cyclic adenosine monophosphate is able to activate different important molecules like transcription factors. Activation of transcription factors, and this regulates gene expression, and gene expression finally regulate activation and production of different enzymes. Termination of action. As we have alpha subunit bounded with GTP, this GTP slowly hydrolyzed from alpha subunit. So with help of water molecules, a lot of water molecules present inside the cell, we have hydrolysis. And we have again alpha subunit bounded with GDP molecule and phosphate. This part, or this complex or GPCR receptor, can be regulated by two ways. The first way, as we have binding of the ligand to this GPCR receptor, and ligand has low affinity and very quickly dissociates from GPCR receptor, this terminates action of this ligand. But if ligand binds for a little bit longer, we have another mechanism with help of beta-arrestin kinases and ATP beta-arrestin and kinases. Sorry, not with beta-arrestin, beta-adrenergic receptor kinases. We have phosphorylation of beta receptor, let's say, and after that we have attachment of beta-arrestin, and this causes internalization of the receptor, and as a result, less amount of receptors are present on the cell membrane.